Yeah, hello everyone. I'm not a chemist, so if I say anything chemically incorrect, please don't crucify me. I'm trying my best with all the terminology. Yeah, this is a project of collaboration between some of the mathematicians at UCLA with some of our friends in the Department of Chemistry, specifically with Professor Ann Andrews and with Cassie Yang, who's at Columbia. And well, let's just get right into the story of the project. All right, so how did we come about working on this project? Well, SciLex data can be very large, so Anne asked Andrea to help her look into this, because Andrea has a lot of history with working with large data sets. The toy data set that we were given was had a total count of only around 200,000, and it was about 5,000 sequences. As you all probably know, this is a very small SciLex data set. We were told to search for these so-called black swan sequences, potentially low-count sequences with interesting and novel chemical properties. So we decided our goal was going to be to determine some way to robustly search through the SciLex data to try to figure out where these black swan sequences could be hiding. This is the flowchart that we developed for doing that. We're going to be discussing the two middle sections here. The first thing that we wanted to do was we were handed a file that had a list of sequences and a list of counts. So we wanted to turn that into more data so we could have more access to some stuff to play around with mathematically. So the first thing that we ended up doing was we were recommended that GMFOLD is a great way for looking at potentially searching for properties. So we wanted to build a in-house version of that, like a secondary structure computation tool that allowed us to do these high throughput secondary structure computations and hopefully pretty quickly. And then we would couple it to some, I say machine learning here, we would call this in applied math classical machine learning, because we aren't using any deep learning techniques. We just use some clustering methods, some other sort of machine learning techniques that we'll talk about later, to sort of try to search through this data set to find sequences that have potentially useful properties. So first, let's talk about, this is the text of what I just said. First, let's talk about some folding algorithms. A lot of what GMFOLD is based on is this original Zucker-Siegler algorithm from 1981. GMFOLD and UNA-fold use this as the quote-unquote gold standard. And recently, in 2023, there was some open source Python-based code to implement just this standard Zucker-Siegler algorithm. GMFOLD does a bit more under the hood than the standard algorithm, but it is a bit less easy to work with. It's not in Python, and it's a bit the code as it's written is a bit more obtuse. So we started with this Python-based open source code, and we were trying to see what we could do potentially to speed it up, because when you're doing high throughput, every second counts. So we used some advancements in the idea behind these folding algorithms is that you're trying to find matching pairs, like so you want to match your T's with your A's and your G's with your C's, because spoilers, we are only at the moment considering the standard Watson-Crick base pair interactions. There is future work for developing this for non-canonical matching, and we do have some ideas in that front. But at least for now, we want to find like some sub-pattern in this large matrix. What is this matrix? We have a green box wherever we have a base pair and its corresponding Watson-Crick counterpart. And what we noticed is that in the Zucker-Siegler algorithm and in GMFOLD, you'll get penalized for having a lone base pairing sitting, like if there's no nearby base pairing, you get penalized with some really high energy value. I believe the number was 1600 kcals per mole, which when each base energy value is around 10 kcals per mole or less than 10, you're never going to be able to subsidize this like energy cost of a single isolated base pair interaction. So you'll always end up seeing these two-level stacks come up in your structure, so we can just filter out all of these edges against only those edges that come in these like pairs of two-layer stacks. And we can just visually see how much of a reduction in edges that we would have to search over just from this one small graph filtering approach. So what we did was we wrote a SecFold 2.0. Again, it's based on the original SecFold. It's currently DNA specific. That can be modified for RNA, and we are working on that. And it's based on the original superscript algorithm. We also did some hash efficiency upgrades to what was done in SecFold, like we eliminated some redundant computations, and we attached this graph matching approach. And there were some incorrect computations of phase energies that were done in that code, so we fixed those up. And we got a pretty good reduction. Both in terms of absolute time computation, but also in terms of time scaling. So just adding the graph matching and cache efficiency, you can see that as you increase your length of your fold sequence, we like for 400, for example, it's about 110 seconds on our SecFold 2.0 versus the average being 175 seconds. So that's like a roughly 50%, 40% time improvement. That's not true. 33% time improvement over the existing algorithm, and as you go on, as you increase the length of your sequence, your time improvement will get better, right? We have our log slope is 3.5 versus the 3.7 of the original algorithm. However, there was still some issues. I haven't talked about how it agrees with MFold yet, right? And that's really the key. Making sure that this algorithm is useful. SecFold originally doesn't super agree with MFold too well. SecFold 2 qualitatively agrees better, but it still felt like we were missing something. So we wrote what we were calling GMFOLD, where we widen our search space because we were using a more robust searching algorithm than the Ziegler-Stiegler one. This allows us to eliminate secondary cache, so we actually are saving about 50% of your memory capacity of the algorithm, and we still include all our old functionality, and we were actually able to get reliably matching, if not very nearly matching structures to MFold. However, there are a few issues. This alternative to MFold is still kind of alive because we haven't fixed some issues yet, but it's getting there, and we have plans to fix these. We're not yet able to handle varying salt concentrations at the moment. We are using some motif energies that were measured at 1 molar sodium chloride. There are algorithms to fix this that we are working on implementing at the moment, so that's not a big issue. We only have limiting coaxial stacking stabilization energies implemented. That's just because going through the literature values to find all of those energies, we have not yet completed that. And we are currently restricted to canonical base pairings. As I said, we do have techniques that we have been developing for expanding to non-canonical, but the canonical base pairing is, of course, the easiest to work with. So that's why we attack that first. And we are enforcing the stacking criteria. I do have an example as to why this might be an issue. Like, I know I was very convincing with saying why it's so obvious. But yeah, in this GMFold algorithm, we were able to match the structures that we get from MFold very nearly, and at least a whole lot better than SecFold did, and even better than the SecFold 2 algorithm. Let's look at some actual structures, because staring at numbers is kind of, even I, as a mathematician, I don't love staring at numbers all day. So we look at some pictures. As you can see, our SecFold, and okay, it didn't cut off. We have SecFold on the left, our SecFold 2 is in the next column. Then we have our graph matching fold, GMFold, and the standard MFold implementation. So as you can see, this is kind of basically what happened across our data sets. Is SecFold and SecFold 2 were very similar, and GMFold and MFold ended up being very similar. However, we do pay a cost because we're using a more robust searching algorithm. It is taking more time to do GMFold, but it turns out that it's not too much more time. I'll tell you why the non-canonical base pairing is kind of an issue. So you can see that MFOLD suggests that for this particular Dioplan aptamer, we're going to have a GT interaction happening here at base pairs 12 and 32. And without this, if we'd never found this in our algorithm, that would leave this GC at 11 and 33 to be an isolated edge, right? And as we said, the isolated edges are bad. They're penalized very, very heavily. So when our GMFOLD and SciFold 2 end up looking at this and seeing this isolated edge, they throw it out. So we only have this small, instead of this whole zippered region, we have only a small stack region stabilizing this sort of binding interaction. So it turns out that our algorithms find that it's more favorable to not have that zip region at all. And you can see that this might be a bit of an issue. We are currently working on ways around this, and we do have some that seem pretty promising. But it was good enough to at least start working on the second step, which was using our machine learning classification to try to search through some select space. In order to do machine learning, you want to have your data embedded potentially in some high-dimensional space. You want to have a lot of descriptors to look at, because then your algorithms have more information about it. So we looked at two sets of descriptors. Technically, it looks like three, but these two sets of descriptors end up being identical: the structural matrix and the small-scale path. The small-scale paths are a mathematical tool that allows you to encode all of the structural information on single-length sequences with non-intersecting edges, right? We don't consider pseudonauts in the super-Stiegler algorithm, so you never have intersecting edges of your secondary structure graph. So these multi paths have been shown to be equivalent information to this folded secondary structure. And this allows us to get some vector, which you see as describing kind of the height at each face. So as we increase along edges, whenever we see a 5' side of a interaction, we take a step up. Whenever we have no interaction, we take a step sideways. And whenever we see a 3' side of an interaction, we take a step down. So you can imagine that each of these heights each of these faces are sitting at a different height, and we're just recording the heights here. So now we have some vector which is describing exactly what's happening in the secondary structure. And then we have a different type of structure classification called this bag of faces. This comes from more machine learning type literature. It comes from the bag of words used for language processing, where instead of just writing down your face energies and trying to record the entire structure, you just record how many of each type you get, and then you'll be able to have some sort of classification based on the similarities of this distribution. So as you can see, we record separately in a vector how many of each, not only just type of face that we have, but the specific energy of that interaction. So hopefully we're encoding some sort of energetic, more complicated energetics of the molecule than just, or of the aptamer than just the total delta G. We get some more detailed information there. So now let's do some clustering. You can think of this plot as, it is a t-stochastic neighbor embedding. You can think of it as just slamming our high-dimensional vector of those. We had the vector of these heights, and just slamming it down into 2 dimensions, and this is what like the splats come out as. And you can see that the highest count sequences from our select screen seem to be in these isolated sort of clusters, apart from these like massive chunks of the rest of the data. And as you would expect, and as you should expect, any sequences that are nearby each other in this low-dimensional embedding will still have identical structure. So this is our first tool in our analysis. We did this, we run a clustering algorithm to segment this data into sequences that are similar to other sequences. And this allows us to accurately get sequences with similar secondary structures to high count sequences. I'll quickly say here the computation time of our GMFOLD algorithm. We were running this on about 5,000 sequences, and on just my colleague's laptop, like a few years old at this point, it took under 3 minutes to compute secondary structures for all of these aptamers. These are roughly length 50 aptamers. It takes 3 minutes for 5,000. So that translates to, if you had 1,000,000 sequences, that would be about 10 hours. And this is just, of course, running on not the highest computational efficiency. And this algorithm is very easy to parallelize. So you should expect that computation time to be cut down significantly if you have more computing resources. So how does the phase energy come into this picture? Whenever we look at these sets of high count sequences, when we look at all of the identical structures, our next step was classifying their phase energies to see how similar energetically they may be. This is all heuristic. We have not done there's no lab work done yet, so we don't actually know the energetics. So if we look at how these descriptions might look a bit vague, but what's going on here is that in each of these figures, this is for one type of structure. And these are the different types of possible energetic configurations with the number of unique sequences with each energetic configuration listed above. So what's happening in these shaded regions are the regions that contain high count aptamers. So what's very surprising is that the high count aptamers were always in the same, always have the same energetic configuration as the most common energetic configuration of their given shape. So we thought this was very surprising, and this may be an interesting place to mine for other useful aptamers, because there might be something going on with this particular shape and energy configuration. But you might have missed something with the amplification step, like the amplification step might have favored some aptamers over others, not for any particular chemical region or chemical reason, but for a chemical reason, but not for the chemical reasons you want to be filtering for. So this is the sort of end of that part of the process, is that we then were able to cut down those 5,000 aptamers. We were able to create an algorithm that outputs the aptamers from each of these different regions that should share the same energetics and the same secondary structure as the starting aptamer. And we fed these off to our colleagues, and they were pretty happy. So our conclusion: we did develop a new Python tool. It is well, it will be open source when the paper is out. It is on GitHub. So it will be an open source Python tool for computing secondary structures with a few novel improvements that haven't been seen before. It allows high throughput calculation of secondary structure. And taking in data from a select screen, we can use these clustering methods to better understand the space of aptamers and identify low-count aptamers that have identical secondary structure and that may share energetic properties. And I don't know the conventional methods, but I was told by our collaborators that this statement can be correct. These should not have been easily identified with conventional methods. And further analysis of these aptamers are needed to see if our hypothesis that they may share or they may have interesting chemical properties, whether this is actually true or not. All right, and I'd like to thank you all for listening. Thank you to the organizers of the conference and to our funding sources, or at least those sources that were funding us at the time that this project was.